Okay. A system that can't self-propagate, of course, in other words, isn't autocatalytic, cannot perpetuate itself. And this system that spontaneously emerged had to also be able to evolve adaptively to explain how we end up with the complexity of life that we're familiar with. So what this first evolver was is a fascinating problem. And that's really what I've been pondering for years, long before I formally entered the field. So I think you're probably aware, but I'll just quickly review that there are multiple proposals out there for what this first evolver was. And I'm just going to quickly review them in a simplified form. So where that's not advancing, there we go. So of course, the oldest, so I'm having difficulty here getting my windows positioned. Give me 1 second. You all know it can be tricky. So the the oldest class, I would say, of theory of thinking scientifically about the origin of life. Imagine that the first living thing had to be a cell or an individual of some kind that emerged spontaneously and was able to grow and divide. And of course, this goes back to Oparin and theories about the coacervates, theories about lipid droplets, and so on. And these ideas basically, imagine that spontaneous chemistry would generate building blocks. These building blocks would self-organize by physical means into these units. And some of these units could be able to self-propagate and evolve adaptively. Now, in some sense, a combination of this strand of thinking, but also the end of it in many ways, was the Miller-Urey experiments, which showed that some of the key components of the supposed structures, amino acids, and by implication then proteins, would be expected to arise spontaneously on the early Earth. So that was, a very highly publicized result, published in 1953, that earned a Nobel Prize, and people were, in one sense, the motivation for that was, where does the stuff come from that could self-organize into these droplets or coacervates or whatever? But ironically, I think it actually ended up marking almost the end of this approach, for the simple reason that within about a month of the publication of this result was the publication of the structure of DNA. And so the publication of the structure of DNA and the resulting studies into how living cells turn DNA into their complex mechanisms of self-propagation shifted the field's attention very much from where does the stuff of life come from to where do the mechanisms of life come from. And the mechanisms of life came to mean the genes. And so in the wake of this work, the attention of the field shifted very dramatically to not asking about imagining that the first evolver was a droplet, but imagining that the first evolver was a nucleic acid. And with the discovery of catalytic RNAs, it became narrowed to the RNA world idea that it must have been a catalytic RNA that was capable of self-propagation. Now, I don't know where you all stand on this, but I would say from my assessment, the pure RNA world hypothesis kind of fallen out of favor because it has a lot of serious problems. The first problem is long RNAs don't form spontaneously, very unstable molecules. You also have the problem that this first self-replicator would only be a self-replicator if its complement were already in the environment. So where would the complement come from? So you now got to explain two things arising, either two self-replicators or a self-replicator and its complement. And then you have the problem of the error threshold that, it's not good enough to promote the self-replication of a sequence, the replication of a sequence. You also have to do so with a low enough error rate that on average, you make at least one copy that is faithful. And if you don't do that, you can't evolve. in a progressive direction. So for all these reasons, the straight up origin of life gene first approaches have sort of fallen away, I think. People have tried to kind of rescue it by imagining not single RNAs, but populations of RNAs kind of captured in some kind of individual, a protocell. But of course, then you've got to explain both the RNA and the spontaneous emergence of the protocell simultaneously and spontaneously, because we're looking for a system that could self-propagate and evolve right away. So everything has to come together without any prior adaptive process. And so this model has had problems. I think they've been recognized for a long time. So what's the third strand? Is sometimes called the metabolism first. I'm going to call it the ecosystem first class of approaches, which, and for reasons that I'll explain in a moment. So these are theories that I view the starting point for this as being Gunther Wächtershauser's paper in 1988. I'm just curious, are you familiar with this paper? I could see you if you raised your hand or something. People familiar with it? No. Okay, you should read this paper. It's pretty amazing. Wächtershauser was a German patent attorney. He had some contacts with, through marriage, actually through a relationship with Carl Woese, who was a molecular microbiologist in Illinois. And he developed this very elaborate and insightful theory proposing that life could have begun with the spontaneous emergence of autocatalytic cycles of chemical reactions absorbed onto mineral surfaces. And he was very precise about this. He specified the surface. He suggested the surface was iron pyrite. And he identified some of the pathways that could have allowed for redox-driven chemistry to generate self-propagating slime, if you will, stuck onto mineral surfaces. And what was interesting about his model is that these slimes, based on he claimed, would immediately be subject to some kind of selection. If they could self-propagate, they also had the potential to complexify over time through an adaptive process filtered by selection. So I read this paper, when did I read it? Probably late 90s or something, when I was preparing to lecture to undergraduates. And it really fascinated me, this idea, because it was completely different. You know, life didn't begin with a compartment. It began as a slime on a surface, and natural selection kicked in right away. But I was very skeptical, of course. It just sounded too good to be true. But it spawned a series of ideas built around the idea that what began was not a particle, not a gene per se, but a system of autocatalytic chemical reactions that collaborated together to propagate themselves and had some innate ability to evolve adaptively. And you know, the initial version was this surface version, the surface metabolism model. Other people like Shapiro had the same kind of idea, but they also imagined that they would be spontaneously emerging inside some kind of protocell. So, the idea here was tantalizing to me, and so I should explain now, why do I call it a metabolism, not a metabolism, but an ecosystem? And you'll see, I'm going to give you a quick intro now, and then I'll come back to it later, hopefully. And please stop me if I'm at all making assumptions about what you know or don't know or terms. Please feel free to do that. So, so on the left, I've shown a sort of a classic example of a small chemical autocatalytic cycle. So in this cycle, basically, a molecule of A can use some kind of food in the environment, some kind of flux of energy and resources in the environment to turn into B. B can then take more maybe different kind of food to turn into C, but then C gives rise to two A's. So what that means is that every turn of this cycle, you double the number of molecules. That's an autocatalytic cycle, and they exist chemically, and I'll show you later, they're actually kind of abundant, hidden inside chemical reaction networks. On the right, we have a life cycle of an organism. So, an egg, given that there's food available in the environment or that it's being provided with food, it can mature into a larva. By food here, we would have to include gases, oxygen and so on. Then the larva will eat food and turn into an adult, and the adult can make multiple eggs. And so a life cycle is also an autocatalytic cycle. And in both cases, nothing happens until you have at least one entity present of the one member of the cycle. So you could have an environment flooded with food, but until you add one molecule of A or B or C, nothing will grow. You have to have one of them. Likewise, you have abundant food in an environment. Until you introduce an egg or a larva or an adult, nothing will happen. So there's a kind of a comparability there at the level of the cycle and the two cycles. And yet that expands out into a metabolism can be thought of as an ecosystem of these life cycles or autocatalytic cycles. So if you imagine that within some kind of chemical reaction network, there are potentially many of these autocatalytic cycles, some of which would be activated, meaning that there is one molecule feeding it that allows it to be active, and some of them wouldn't be. These autocatalytic cycles are competing for food potentially. They are generating maybe the same waste, which means they're competing, chemically speaking, for waste. They might be eating one another. So the food of one might be the member species of another cycle. And there's a network of interactions that explains why biological ecosystems can change over time through processes like ecological succession. You take bare rock and gradually it becomes a forest. And so this parallel, as you'll see later, sort of after I give you some empirical results, provides a useful framework for thinking about the origin of life. So I became exposed to this way of thinking, and it convinced me that actually we have made an assumption in the origin of life field that the emergence of the first evolver is difficult and rare. What if it were in fact simple and easy? What if you took almost any environment that was driven out of environment by a flux of food, if you could get the spontaneous emergence of these self-propagating chemical ecosystems that could then change over time through a process that might look like succession and might ultimately look like evolution? How would we know that was happening? And I realized that nobody had really done experiments to assess that possibility. The people had assumed that, yeah, you could make amino acids in a vial with sparks, but they weren't going to do anything if you didn't wait and watch what happened over time. So, coming from an evolution and ecology field, I thought, how would we test this? How would we explore it? And so we ended up designing an experiment with Kalin Betzigian, who was a colleague of mine there, and a very wonderful graduate student, Lena Vincent, has since implemented this experiment for the first time. And it's an experiment that we call chemical ecosystem selection. And so I'll try and explain the design here, the concept. So the idea is we're going to create a vial that is sort of a microcosm of an early Earth. So it contains, now let me just get a pointer going. Hang on, how do I do that? I can't remember it. Can you see my pointer? You can't, can you? You can? Yes, okay, I'm seeing some nodding from Luna there. So it's actually working. But so we have a vial and it contains an atmosphere, and we make it anoxic, like the prebiotic Earth. We have some liquid, we make it, this experiment, it's kind of like seawater. But it contains, in this case, the solutions that would have been created by a Miller-Urey spark discharge experiment. So it contains a prebiotic soup. And then it contains a bed of minerals, and we use iron pyrite in the spirit of Westershauser. So the idea might be that perhaps if this is a simple process creating a self-propagating system on a surface in this model, then some of these surfaces of the minerals would get spontaneously triggered through some kind of rare reaction. They would start triggering some of those autocatalytic systems. But different grains might end up with different autocatalytic systems that could be spreading over the surface of the grain. But how would we know that was happening? Well, the way we would know that was happening was by starting a serial dilution experiment. So basically, we would incubate for a period of time, then we would transfer some fraction of the minerals and maybe the soup too into a new vial that contained virgin grains and fresh soup, so fresh food. So this would dilute any systems that had emerged, and then we would dilute them again and again and again, and only systems that could self-propagate themselves. And in this context, they would have to not only emerge but get between one grain and another grain. Only systems that could self-propagate, that were collectively autocatalytic, would get enriched over time. And if they became enriched over time, and there were multiple variants, the one that would win would be the best at self-propagating in that environment. And so there's a process of selection going on here that basically could result in a system that in generation 1 looks very different than generation N. Even though each generation, the vials have been incubated for the same amount of time. It's just that this has accumulated a history due to these seeds that have been transferred from one generation to the next. And so the hypothesis was that if autocatalytic systems emerged, you would see systematic changes in the chemistry of the solution at the end of the incubation. These systems would have to eat food. In the process, they're going to change the chemical makeup of the solution in some way, almost certainly. So if we were to see systematic changes in the composition of the soup at the end of the incubation, that would suggest that a system had been self-propagating spontaneously. So, and I keep saying, please stop me if I'm unclear, but so what we did was, this is just a recap really, we created a soup that we thought, was a proxy for the kinds of soups that might have been formed on the prebiotic earth. So we used data from prior experiments to select, concentrations of different amino acids and organic acids and so on. We situated it in seawater. We made a point to have the solution be divided into 2 sub-solutions, because some of the energy in a chemical mixture is in the disequilibrium between the components. So we wanted to keep the soup sort of able to express that disequilibrium, and we would make it fresh each generation by mixing the 2 sub-soups. I said we used iron pyrite, that we got natural iron pyrite, ground it up, size selected it, cleaned it to get rid of any oxidized layers. And then we did this serial transfer experiment, but we were very concerned about false positives. And the main false positive to be concerned about is life itself getting into the vial and growing. And so the way we kind of avoided that possibility, we think, was to incubate, to put autoclave the vials at the start of each generation. The first thing that happens is they go in the autoclave, and then they come out, and then they can incubate for in this case, 2 to 3 days. And so then the question is, what do we track? Now, nowadays, we've shifted over to trying to use very sophisticated methods like mass spectrometry to look at what's in these soups and how they change. But in this first experiment, we just used high-level proxy traits, sort of general survey measurements about a solution. So like pH, we also measured minerals, like changes in phosphate concentration, and UV-vis absorbance, just to look at sort of what chemicals are there. And then the question was, would they be changes over time? So let me summarize some results. So, so in this experiment, there were 10 experimental lineages, and they had been, and this is just showing the result after 6 generations of transfer. So that means that, actually the 6th generation, so they've had 5 transfers in their past, and these are the 10 lineages. And as a control, what we would do then is the generation before that we made this measurement, we would set up another set of parallel vials. Is everything okay there? Yeah. We would then set up the same set of parallel vials with the same solutions in case anything had changed, and we would measure those after 1 transfer. And as you can see, this is measuring the phosphate concentration. There's no difference between the experimentals and the controls in generation 6. But at generation 12, the experimentals look different. All of them look different than the controls. So history here, having 11 transfers in your past instead of just one transfer, has an effect. So that's kind of something's changed systematically. And in generation 18, it was even more extreme. So the phosphate that was previously in solution at the end of the incubation is not there anymore. It's either being covalently bonded to something, or it's absorbed to something, but it's not measurable. So we also measured the absorbance of the solution at different wavelengths, which is a measure of just how much organic molecules there are of different kinds. And what we found is, if we standardize to the control, so this is, for each wavelength, we just set the control to 1, and this is the variance around the control. The experimentals at basically every wavelength, this is in generation 18, this is averaging over the 10 experimental vials, they have a lower absorbance. So the organic solution compounds have also disappeared from the solution. So where have they gone? So we had the idea just to take a look at the grains at the end of the incubations. And what we found is that in generation 18, many of the grains had these structures on them, these crystalline fractals, which were very intriguing. And I'll be honest, when I first saw these, I was like, my goodness, those are these slimes. They are living things that have emerged spontaneously. But we now think they're not, okay? What we think they are is that there's just organics, probably, and phosphate absorbing to these surfaces for some reason. And because of their absorbing to the surfaces, when we dry the solution out to look at it into the electron microscope, crystals of salt form on the surface, and they have these fractal shapes. But it does point to the fact that probably both the phosphate and the organics have been absorbed onto the minerals over time in some way. So that was interesting. And what happened in this experiment was we continued it for another 6 generations. They looked at generation 24. And what was weird was in generation 24, everything went back. It looked like it had gone back to the original condition. They looked no different than the controls again. So we said to ourselves, oh, we probably made a mistake. Maybe we got oxygen in there, something went wrong. Let's do it again. So we did a long-term experiment, and the same thing happened again. Slightly different timing, but what happened in the 10 experimental lineages, the phosphate concentration steadily dropped and then suddenly went back up again. And it was showing you the averaging of the 10 vials here. You can see the gradually the phosphate dropped. We start seeing these fractal structures, and then they bounce back up again. And then they gradually drop again, and then they bounce back up again. And then it becomes chaotic. So what does this mean? Well, it's, there's many possible interpretations of this, and we don't know the answer. One interpretation, and this is sort of a generous interpretation, is that we did have some sort of chemical ecosystem emerge that could feed on disequilibria in the soup, grow, but it couldn't persist when it ran out of food. So the concentration of this system increased over time, but it got to a point where there was so much of it that it ate all the food, and then it broke down and dissolved. That's one interpretation, sort of ecological interpretation. But it was tantalizing nonetheless, and suggested that this kind of experiment could reveal interesting information about the spontaneous emergence of these chemical ecosystems. So, so what we're doing now experimentally, COVID really unfortunately disrupted this experimental program significantly. Additionally, we had some instrument issues, but we're now moving to using more mass spectrometry, LCMS methods to really see what chemicals are present rather than just look at phosphate or pH or redox, whatever. We're also looking at different soups. We've started making soups that are much more, so the model, the Miller-Urey soup, the heterotrophic model imagines that abiotically, a lot of carbon is fixed into organic molecules, and then these incipient first evolvers feed on the organics. But the other possibility is that actually these incipients, the first evolvers, feed directly on CO2 and inorganic food sources. And so we're also making some autotrophic soups, so basically inorganic. And we are finding organics emerge, but we haven't yet seen one of these systematic patterns over generations. We're also doing experiments that really try and push the concept to see if, really trying to assess whether the changes can be interpreted through a lens of adaptation. So for example, we can do experiments where we change the environment and see if different lineages sort of remember what environment they were in and sort of become adapted to it, and do less well when we challenge them with a different environment compared to a set that have got experienced at that environment, if you will. So that's what we're doing now experimentally. But what happened was, I had this intuitive sense about these ecosystems and did these experiments. And then I really want to make sure that the underlying theory is sound. So we've actually done quite a lot of theoretical work, which is what I had intended to talk about for the rest of today. But if you like, I could pause now if any questions about the experiments. Would that be useful? It's up to you. Joaquin, do you want to advise me?
Can you, yeah. So perhaps yes.
Okay, that's fine.
I don't know, are there any questions from people here? If not, we can carry on, and if they arise during the rest of the talk, we can move on, or up to you.
I'll pause for five seconds, and then I can keep going if no one has questions.
I don't know, I'll keep an eye on the chat just in case. Maybe not.
I have the chat window open too.
I might come later, but just in case. If not, we have some time at the end.
Okay. Okay, so I'll go on and tell you about the theory then. Okay. So it's all very well to say these chemicals ecosystems spontaneously emerge and can evolve, but how can a system that doesn't have a genetic code, how can it evolve? And so we wanted to approach that sort of formally and actually, make sense of this. So the first thing we have to really understand is, well, what are the similarities between a chemical reaction network driven by a fluxus of food and a biological ecosystem? And so we started by just mathematically analyzing autocatalytic chemical cycles. And this is a very simple one. This is just a two-member cycle. And remembering that unlike life cycles, chemical reactions are reversible. You can't go from a larva back to an egg, but you can, almost any chemical reaction can go backwards. And so we wondered whether this would change the behavior in an important way. And so what we did was we simulated, well, actually we mathematically analyzed and then also simulated what happens if you put autocatalytic chemical cycles into an environment where they receive a flux of food, and the food and the waste and everything is being washed out at a constant rate. What happens is the member chemicals of the autocatalytic cycle, they grow to some carrying capacity. And I don't know how many of you know any ecology, but this is exactly what biological species do. So even though the chemical reactions are reversible, because food is constantly flowing in, effectively you're driving it in one direction. And so that's why you get logistic growth to a stable value. Okay, that's a single cycle. But what happens if you have two cycles that interact with one another? So what we did was we set up different kinds of networks which represented different ecological conditions. So we can have competition where we have two cycles competing for food, and we can get different outcomes. They could coexist in some conditions, or one of them can exclude the other. If one is a stronger cycle, it could exclude the weaker cycle. We could also set up predator-prey dynamics where one cycle produces and contains a member that the other cycle has to eat for food. And we can get extinction of the predator, we can get coexistence, but we can also get complicated temporal dynamics, depending on exactly the parameters of the rate reactions in these chemical reaction networks. So what this leads, and there are other cases too, mutualism and inhibition that we could demonstrate. So it really does seem a good metaphor, at least, a good analogy to think of chemical reaction networks that contain multiple autocatalytic cycles as these ecosystems. So then the next question to ask is, but how might they change over time? So in order to evolve, you have to have some mechanism of change, and a mechanism of change that is heritable, you can remember, the system can remember. These have this ability because of this necessity for a seed. So imagine you have an autocatalytic system that's been triggered, where these lowercase symbols represent chemical food in the environment, and the uppercase letters represent members that have these reactions. And I'm only showing the forward direction, but they may be all reversible. So as long as you provide little a, little b, little c, and little d, this cycle will make more and more of itself, and it will generate more and more waste. And that waste, though, might be a potential food for another autocatalytic cycle that hasn't yet been seeded. So this yellow cycle wouldn't be there unless an E, an F, a G, or an H of the uppercase kind emerged. And it could emerge due to a very rare local reaction, like a super low rate constant. It could be brought in on an asteroid. It could flow in from another environment which has different catalysts, so different things are happening. Once the yellow is triggered, though, provided that the food it needs is present, it also will now be sustained. So the system, in a sense, could remember that a seed had arrived at some point in time to this environment. So there is a basic kind of ability here. So if real chemistry has multiple autocatalytic cycles, then there's the potential for this sequential addition of autocatalytic cycles. So we wondered whether real chemical networks have those properties. I guess this is just a way that I'm not sure. Here's the way that I think about the overall hypothesis I'm presenting to you. It's basically that there's food flowing into an environment, local environment, and it might trigger some kind of autocatalytic system that just emerges spontaneously there and can sustain itself. So now the local systems are not at equilibrium with the environmental flux. They're kind of chemically different than the environment around them. And there might even be two different kinds of autocatalytic systems that might be triggered by chance, one or the other. Then what might happen is a seeding events might occur where something drops in from the outside, activates new cycles, so that in turn might drive some old cycles extinct or might allow yet more cycles to emerge. And over time, we can imagine these things complexifying or changing, becoming more and more out of equilibrium with the environment. And I'm not going through it here, but we can also imagine how selection might act on this. Some of these systems might be more tolerant of, let's say, a period of time where there's no food, or might be better at spreading onto more mineral surfaces. Those ones will tend to perpetuate themselves, whereas the ones that are less good will disappear. So some kind of basic kind of selection could kick in, again, without any kind of genes. The memory is not in genes. The memory is in which autocatalytic cycles are activated. If you're interested, Sass-Marie has called these cycles chemes, like genes chemes. They're like chemical memory holders. But we wonder, do real chemical networks have these properties? And so what we've done recently, and I tend to work in a preprint that we have produced about this work, we analyzed two chemical reaction networks. One of them is clearly abiotic. It's a reaction network of radiolytic reactions. The other one is biological. It is the biochemical reactions metabolism, as inferred, as might have been true for the last universal common ancestor, Luca. So we asked the question, do these two chemical reaction networks have an organization that would allow for these kinds of dynamics? So starting with the abiotic one, what we can ask the question is, let's pick some food. Let's just decide what it is that might flow into the system. So let's say we pick methane and hydrogen and NO and FeS2 and visible light. This abiotic network has a lot of electromagnetic energy sources in it. So what would happen? Well, nothing would happen. They would just flow in and that's it. So we then asked, well, among the other 277 entities in this network, it's a pretty small network. If you added one molecule of one of them, what could you trigger an autocatalytic cycle that would regenerate the seed? And the answer is you could. There are, oh, I beg your pardon. Yeah, I'm sorry. So this just shows that the food entities are already present. So those will be there. So we call that tier 0. Those are the things that would be there without adding any seeds. Then we can add seeds, and it turns out there are 12 different chemicals that you could add, chemicals or electromagnetic sources you could add, that if you add them, it triggers an autocatalytic system to emerge. And this autocatalytic system is not just there, those 12, it contains 91 different entities. And within this system, there has to be, it couldn't maintain itself unless there were autocatalytic motifs in there. And we have an algorithm that can find those autocatalytic motifs. Turns out there are lots. So we can ask it to find the smallest one, just to take a look at it. And this is the smallest autocatalytic motif, autocatalytic cycle in here. And this cycle feeds on visible light, NO and CH4. And each of the dashed lines show the reactions. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 reactions. Everything in a red box is a member of the cycle. It's produced during the cycle and needed for the cycle to continue. And then there's 2 waste products, infrared light, and I can't actually read that on my scale, but whatever that says. My glasses aren't good enough at this scale. So there is a seed-dependent system. So if one of these molecules blew in, one of these 12, this system would now be established. The system would remember that it was in this state. Well, of course, now there's more chemicals in the environment. There's 91 additional chemicals. Some of these chemicals might be the food of another system that hasn't yet been seeded. So is there a second seeding event that could happen? Is there a tier 2 system? And so it turns out there is a second level that there are 13 chemicals that can seed, and those chemicals seed a system that is actually only just slightly bigger than those 13. And for those of you who know chemistry, this is basically the Formose reaction, which is a well-known autocatalytic cycle. And that's actually as far as we can go in the abiotic network. It's a kind of a small network. So now let's look at the biotic network. Oh, and there's the Formose reaction. It's a very small, simple cycle. So in the biotic network, we picked the food on the left, just inorganic food that could provide phosphate and sulfates and so on. And they actually react with one another, so that if you don't add any seeds, you have 30 entities present in the system. We then looked at all the other chemicals one by one to ask which individually could seed a system that was self-sustaining. We call them seed-dependent autocatalytic systems, or SDASs. And it turned out there were 3 of them that could do this, 3 sets. So there are 267 chemicals that all seed the same system. There's 34 chemicals that seed a different system. And there's 3 chemicals that seed a yet different system. But they're kind of nested, that when you see the things that seed click 3 seeds a system that contains all of the chemicals that seed click 1 produces 2. But what's noticeable is that each of these seed clicks contains molecules of different complexity. So seed click 1, you can see that click with acetylene or pyruvate or glycolaldehyde, a lot of very simple molecules you can seed that with. To seed click 2, the simplest molecule in there is a pyrimidine. And the simplest molecule in the seed click 3 is actually NADH, so a pretty complicated molecule. So a priori, it seems like we should imagine that seed click 1 would be the one that was seeded, because it's just much more likely that a seed of that simplicity would come along. So if any of those 267 seeds were to come along, it would trigger a system containing 301 entities. And then what could happen? Well, it turns out that there are 6 singleton seeds that you could add that would trigger a tier 2 system containing 56 entities and actually starts making pyrimidines. And then there are 3 seeds that could make 1,057 entities. You can actually get there in smaller steps if you allow not one seeding molecule, but 2 simpler molecules to sometimes do the same seeding as one complex molecule. So basically the 2 networks, both the network of life and the network of non-life, have this ability, at least topologically, to go through successive steps of accreting complexity if activated in an environment given a flux of food. So one thing that I wanted to highlight, though, which is really interesting to me, because we know a lot about the chemistry of the biotic system, we can sort of look and ask the question, well, what capabilities emerge with these higher tier systems? And it turns out that when we look at the tier 3 system, it interestingly contains a lot of molecules that actually can catalyze reactions of the earlier systems. So, for example, they produce NADH or FAD. These molecules can aid in getting energy from the environment, getting phosphate from the environment. And so once the tier, you might imagine that as you add tiers, because the number of chemicals you're producing has gone up, the concentration of each chemical must go down because they're getting the same flux of food. But if the system, as it adds these tiers, gets more efficient at turning food into, let's call it biomass, into its own stuff, then it doesn't follow. It could be that as you add more tiers, you start using the food more efficiently, and the whole biomass can increase, not decrease. Even each individual chemical's biomass can also increase. So it suggests that there's a way in which, over time, chemical ecosystems might get fitter. They might get better in some objective way. So I want to watch the time. I don't want to talk too long. I want a lot of time for questions. I'm going to just whiz through just to give you a feel, just to end, to say, we're also doing models to try and understand what happens in a spatial context, because selection has to happen in a spatial context, I think. And so we've been playing where we've been able to simulate these chemical reaction networks on mineral surfaces where we allow diffusion across the surface. And we can get some interesting dynamics emerging. I'll just give you, just to give you a hint. For example, we can get kind of selective-like dynamics where, depending on the environment, different characteristics of cycles can be favored. So we set up cycles here that mutually inhibit one another, so they compete basically. And it turns out that being a fiercer cycle, having higher rates of reaction, is always better. Being a faster cycle, a cycle that diffuses quickly, is often beneficial because you can get to empty food patches quicker. But there are conditions when actually being too fast is bad, because you kind of flow out of the system too fast, and you can't kind of establish yourself. We've also found that you can have, depending on a temporal scenario of turnover in the environment, you can have a case where sometimes a fast reaction network is favored and sometimes a fierce one is favored, depending on the turnover of the environment. So you can start seeing selection kind of acting at the level of autocatalytic cycles. So again, no genes. So it's purely analog inheritance, no digital encoding of anything. And then the last thing we've been looking at is, if it's true that life began evolving in this analog mode with these chemical reaction systems changing, moving through chemical space. How would we ever get genes? My email is at the bottom of that screen there. You can see it. It's pretty easy one.
Okay.
So happy.
Yeah, that's really right. Yeah.
Yeah.
Probably when we, or whenever we post this to YouTube or stuff like that, that's where some more questions arrive. So that might come to and
I'm also going to make a suggestion that you might want to look up Lena Vince. If you're big in social media, Lena's got a great presence on, particularly on TikTok right now, talking about astrobiology. So you might want to,
I think we have her on the radar. We travel. I think she's doing a really good job, and I think it's we've been talking about that actually in some of our meetings, that it's good to see some people trying to do outreach through perhaps not some traditional platforms of these kind of things. I guess that Twitter, for example, is a more normal space to share science. But it seems like she's doing a really wonderful job.
Yeah, I'm very, very happy with what she's doing. It's great. So, good.
Yeah, well,
okay.
Those,
thank you for the invitation. It was
of course.
I will stop sharing.
I think from our side, we just wanted to mention that so this this will be the first of a few talks. So in November, mid-November, mid-December, we'll do 2 more. As general announcements from the society, I think all pretty much we have to say is that we have just started a volunteering program, so people can become interns for the society and help us in this. Obviously, we all volunteer for this, so we're just recruiting volunteers right now, and we also have a exciting announcement to make. For those that have been following us around, we see some we recognize some names here. We finally, after a year working trying to get it to turn into reality, we now have a website, which is astrobiologysociety.com, and it will be formally announced at some point this week through our social media normal channels, and as for that, it's still early stages of development, but we are quite happy that it has finally happened, and will be hopefully a better means of sharing what astrobiology is and sharing wonderful stuff as the one that's been done in your lab, and so many others that are working really hard to give us a better understanding of what are the origins of life, what's life, and if we can find it either somewhere in the universe or even Earth. This gives a really good insight into that. So any other questions from either people in person or anyone online that's something like that? Well, thank you so much again, David, for coming today.
I just thanks for the invitation.
See you around.
Thank you.
Okay, bye.